Hi everyone, good morning. Today we're going to talk about the transfer RNA structure and function. As we all know, transfer RNA is a very small RNA molecule and it plays a crucial role in the translation of genetic information from messenger RNA into protein. It acts as an adapter molecule. It matches very specific amino acids to its corresponding codon on the mRNA during protein synthesis. And here we're going to look at the detailed structure of this transfer RNA molecule. Before we get into it, let's just look at the yellow box on the side. It's a very important feature mentioned here about the genetic code, which is degenerate and redundant. What do we mean when we say that the genetic code is degenerate and redundant? Most amino acids, as we know, are coded by multiple codons. And we're going to look at one specific hypothesis over here, which is the wobble hypothesis. So, in any mRNA sequence, the first two nucleotides of that mRNA codon are essential for anticodon recognition, while the third nucleotide, it can differ. So it can change, but the amino acid added to the protein structure will not change. The exception, however, is methionine, which is coded for by AUG, and tryptophan, which is coded for UGG. Both of these amino acids are coded by only one codon. So I'm going to repeat that again. There's methionine and there's tryptophan. Methionine is coded for by AUG and tryptophan is coded by UGG. Only two amino acids that have only one codon that codes for them. There's no wobble in this one, in these two, sorry. So coming back to the primary structure of transfer RNA, the primary structure is a nucleotide sequence. This is really important, typically around 70 to 90 nucleotides in length. So that information there, that it's 70 to 90 nucleotides in length, that's something very important to remember for the examination because this key detail will pop up in the questions. Nucleotides. It's a string of ribonucleotides A, U, G, and C linked together by phosphodiesterase bonds. The sequence specificity, each transfer RNA has a unique sequence that is specific to the amino acid it carries. The 3' all transfer RNAs have a conserved CCA sequence at the 3' end where the amino acid is attached. This is very important. This can come up with questions. There will be a question like, what is the sequence at the 3' end of this transfer RNA molecule? And you'll have to know that that sequence is CCA. This is very important to remember. The sequence of the transfer RNA molecule at the 3' end is CCA. And what's the anticodon? The anticodon is a specific sequence of three nucleotides that is complementary to the messenger RNA codon. Here below, I've put in primary structure of the transfer RNA molecule. Don't be fooled, it looks a lot like the messenger RNA, but in the primary structure without the bonds, it looks just like this. Okay, now let's move on to the secondary structure of tRNA. The secondary structure of tRNA refers to this folded clover leaf shaped structure that you can see at the bottom right hand of the screen. And the transfer RNA adopts this molecule due to internal base pairing within the primary structure. So the structure includes several regions. You can see the purple portion as the acceptor stem, the maroonish region as the D loop. The anticodon is where the black and the blue loops are drawn in the diagram. You have the variable loop and the T trident C arm. So just looking at the acceptor stem, it's very simple, double-stranded region formed by the pairing of the 5'3' ends of the primary structure with a single stranded 3' end that has the CCA sequence. So you can see that the CCA sequence is there, the 3' end. That's a very important thing to remember. It has a hydroxyl bond. That's where your amino acid is going to attach before it joins the polypeptide change as it is being translated on via the messenger RNA. The D loop, that's the maroon area on the figure. It contains the nucleotide dihydrouridine and forms a stem loop structure. So that's your first stem loop structure that's being formed. and it's called the D loop because it has two dihydrogen nucleotides there. That's very important because in the question stem sometimes they might want you to identify that they're talking about a transfer RNA structure or a specific transfer RNA structure. And the only clue they'll give you is probably the CCA sequence, and probably they'll mention, dihydrouridine. They won't mention the loose, but they'll say it contains a nucleotide, it contains dihydrogen in its structure. And moving on to the anticodon loop, the anticodon loop obviously contains the anticodon. It's a sequence of three nucleotides that pairs with the messenger RNA codon during translation. That's the easy thing over there. The variable loop, that length, the length and sequence varies between different transfer RNA molecules. And the TC loop, it contains a sequence thymidine and pseudouridine and cytidine and forms another stem loop structure. So this loop um, is also very important when it comes down to identifying that, like maybe the stem or the stem of the question is referring to a transfer RNA molecule. I mean, it's quite easy to just say, right? Like, this is a transfer RNA molecule, so what's at the 3' N? What they'll probably do is that they'll mention these particular nucleotide sequences, and that should, ring some bells. The sequences that they'll mention, are um, thymidine, there's uridine, and cytidine. So that's very important. Dihydrouridine is in the D loop, thymine, pseudouridine, and cytidine, they're in the TC loop. So looking a little bit into a little bit more detail of this structure, um, it's located the acceptor stem. It's located at 3' end of the transfer RNA molecule. It consists of about 7 to 9 base pairs, and it's formed by pairing of the 5' and 3' ends. We've already gone through that. The 3' end has a CCA sequence where the amino acid attaches. The D loop contains a conserved dihydrogen D residue and it forms a stem loop. The anticodon loop contains the anticodon, a sequence of three nucleotides that base pairs with the complementary codon on the mRNA positioned opposite the acceptor stem. The variable loop located between anticodon loop and the TC loop, its size and sequence can vary between different tRNAs. And the TC loop contains a sequence thymidine, pseudouridine, and cytidine. Very important, thymidine, pseudouridine, and cytidine. And it forms a stem loop structure. So the tertiary structure of the tRNA molecule is three-dimensional. It has an L-shaped form. It's stabilized by various interactions, including hydrogen bonds and stacking interactions between bases. Again, I think um this is really important. Once again, I've just circled the CCA portion. And again, the 75 to 90 nucleotides is super, super important to remember that if the polypeptide mentioned in the stem has 75 to 90 nucleotides, think transfer RNA. Secondary structure cloverleaf form anticodon end is 3' opposite 3' amino acyl end. All transfer RNAs, both eukaryotic and prokaryotic, have CCA at the 3' end. So that's another important piece of information there, that both eukaryotic and prokaryotic have the CCA at the 3' end, along with a high percentage of chemically modified bases. The amino acid is covalently bound to the 3' end of the tRNA. T-arm contains the ribothymidine, pseudouridine, cytidine sequence necessary for tRNA ribosome binding. So there you go. That's the function of the T-arm. It's involved with tRNA ribosome binding. The T-arm contains dihydrouridine residues necessary for tRNA recognition by the correct amino acid tRNA synthetase. Now just looking at the differences, the primary structure is linear sequence of nucleotides. The secondary obviously is three-dimensional. The primary structure contains a nucleoside sequence AUGC. The secondary structures, it contains stems, loops, anticodon, variable loop, TC loops, etc. The primary structure determines the identity of the tRNA and its specific anticodon. The secondary structure enables proper folding and interaction with the mRNA and ribosome during translation. Very important differences. So what are the functions of tRNA? The amino acid attachment. The acceptor stem binds a specific amino acid facilitated by the amino acyl tRNA synthetases. The anticodon interaction, the anticodon loop pairs with the corresponding codon on the mRNA, ensuring the correct amino acid is added to the growing polypeptide chain. Peptide bond formation. During translation, the amino acid attached to the tRNA is transferred to the growing polypeptide chain. And here is the reaction. Very simple. That's your the transfer RNA molecule. You need ATP for that, and then the byproduct is AMP plus PPI. The enzyme is amino acyl tRNA synthetase. And there you go. After this reaction, the amino acid is attached at the CCA3- end with the hydroxyl group over there. So the amino acyl tRNA synthetase uses ATP, uh, one unique enzyme per respective amino acid, and binding of charged tRNA to the codon are responsible for the accuracy of amino acid selection. Amino acyl tRNA synthetase matches an amino acid to the tRNA by scrutinizing the amino acid before and after it binds to tRNA. If an incorrect amino acid is attached, the bond is hydrolyzed. Remember, if an incorrect amino acid is attached, that bond is hydrolyzed. And a mischarged tRNA reads the usual codon. Sorry, a mischarged tRNA will read the usual codon, but inserts the wrong amino acid. So a mischarged tRNA, that's very important. A mischarged tRNA will read the usual codon, but it will insert the wrong amino acid. So just mentioning the process how an amino acid is added to the tRNA molecule. The process is called amino acylation, also known as tRNA charging. It is the process by which an amino acid is attached to its corresponding tRNA molecule. This reaction is catalyzed by the enzyme known as amino acyl tRNA synthetases. Amino acylation is the crucial step in the translation of genetic information from mRNA into proteins because it ensures that the correct amino acid is incorporated into the growing polypeptide chain. And here are the steps mentioned below. So the activation of the amino acid will react with adenosine triphosphate to form an amino acyl uh, adenylate amino acyl AMP intermediate and releases pyrophosphate. This step activates the amino acid and is catalyzed by amino acyl tRNA synthetase. The reaction can be summarized as below. So the amino acid combines with ATP, it forms amino acyl AMP plus a pyrophosphate. Transfer to tRNA. The activated amino acid amino acyl AMP is then transferred to the reaction of the tRNA molecule at the adenine of the CCA sequence. This results in the formation of amino acyl tRNA and the release of AMP. The reaction can be summarized as below. So you have amino acyl AMP plus tRNA resulting in amino acyl tRNA plus AMP. Okay, so let's look at tRNA pairing, particularly during translation. So it involves the interaction between obviously the anticodon and the tRNA and the codon and the messenger RNA. The codon anticodon pairing, each transfer RNA molecule has an anticodon that is complementary to an mRNA codon. This pairing follows very specific base pairing rules. Adenine will pair with uracil in RNA. Guanine pairs with cytosine. Inosine, very important, inosine in tRNA can pair with A, U, or C, providing flexibility, wobble-based pairing. Codon-anticodon interaction. Codon on mRNA, a sequence of three nucleotides on the mRNA that specifies a particular amino acid. Anticodon on tRNA, a complementary sequence of three nucleotides on the tRNA that pairs with the mRNA codon. If you look at the example provided on the right side, you have an mRNA codon, which is coding for methionine. As you can see, it has a code that says AUG, so you know that that codes for methionine. And you have tRNA, which is the anticodon, which says UAC. Now, this will ensure that when these two pair together, that methionine is added to the growing polypeptide chain. So the steps during the tRNA pairing during translation. So here you have again the example on the side, which is methionine AUG on the mRNA and UAC on the tRNA. Initiation, the small ribosomal subunit binds to the mRNA. The initiator, tRNA, carries methionine, pairs with the start codon AUG on the mRNA. So a tRNA with an anticodon complementary to the next codon on the mRNA enters a site of the ribosome during elongation. The anticodon pairs with the codon, ensuring the correct amino acid is brought into position, and then peptide bond forms between the amino acid in the P and the A sites. The ribosome moves along the mRNA, shifting the tRNA from the A site to the P site. So the ribosome is moving along the mRNA, but it's shifting the tRNA from the A site to the P site. When a stop codon, so this is when it terminates, this is when the polypeptide chain will terminate. When a stop codon UAA, this is very important. What are the stop codons? There are three stop codons: UAA, UAG, and UGA. I'm going to say it again because it's very important. UAA, UAG, and UGA is reached. No corresponding tRNA can pair with it. Release factors will bind to the stop codon. So that's the next step. What happens when when the tRNA will encounter a stop codon and the mRNA, uh, no corresponding tRNAs can pair with it. Okay, and the release factors will bind to the stop codon, causing the ribosomes to release the completed polypeptide chain. So the release factors, they will bind to the stop codon, and that will cause the ribosome to release the completed polypeptide chain. And again, coming back to wobble-based pairing, I've already told you that this is a feature of the genetic code. It's degenerate and redundant. What that means is that most amino acids are coded by form multiple codons. The first two nucleotides of the mRNA codon are essential for anticodon recognition, while the third nucleotide can differ. This is wobble, but there are two exceptions. That's AUG, methionine, and tryptophan, UGG. They're encoded by only one codon. So wobble-based pairing allows for some flexibility in the pairing between the third base of the mRNA codon and the first base of the tRNA antinucleotide. This flexibility helps explain why multiple codons can code for the same amino acids. Although it's the third base on the mRNA code, it is the first base on the tRNA code. So it's the first base on the tRNA code. So here you have the here I've put down inosine because inosine in the anticodon can pair with A, U, or C in the codon. So here's an example of a wobble. Consider the codon GCU, which codes for alanine. So the mRNA codon, if you look at the example I provided on top in the top right corner, so you have the mRNA codon reading as GCC, although the mRNA codon should be saying GCU, but it says GCC, and the tRNA anticodon is CGI, and over here it says CGG. So in this case, the inosine in the anticodon can pair with the U in the codon, allowing for the correct incorporation of alanine. Understanding tRNA pairing is crucial for comprehending how genetic information is translated into proteins, ensuring the accurate synthesis of proteins according to the genetic code. Now we'll move on to a question. I got two questions just to clear some concepts. So I'll read the question. A specific RNA containing 90 nucleotides is purified for further analysis. It is found to contain high amounts of chemically modified bases such as dihydrouracil, pseudouracil, and ribothiamidine, and its secondary structure arises from base pairing within the chain. Which of the following is the most likely composition at 3' end of this molecule? And I think all of you guys know the answer is CCA. The clues in the question: 90 nucleotides, and they're talking about dihydrouracil, which is in the D loop. There's pseudouracil and ribothiamidine mentioned in the question. Um, and they're also talking about a secondary structure that results with base pairing, and they've very clearly mentioned the 3' end of this molecule, so the answer is CCA. Looking at the second question, there are two mRNA sequences which reads as CUC and CUU trinucleotide repeats. After incubating these mRNA solution containing functional ribosomes and tRNAs charged with various amino acids, it is found that both mRNA sequences produce polypeptide chains containing leucine repeats. This observed finding is due to which of the following genetic principles? So this is because of wobble. And as we said that, like obviously if the um, it's okay to, if there is a little bit of problem on the third nucleotide on the mRNA because many amino acids are coded for by multiple codons, with the exception, obviously, methionine and tryptophan. Methionine is coded by AUG and tryptophan UGG. They only coded for by one codon. So thank you guys. I hope this was helpful.